Hello, my name is LinhChi Ho. I am currently a junior biology pre-med major, and I work at the Arkansas Biosciences Institute in Dr. Fabricio Medina-Bolivar's lab. I do research with another undergraduate student named Cecily Long and a doctoral student named Zebedee Mohammed Hosenpour. The title of my poster presentation is Evaluating Prenylated Stilbenoids from Peanut as Potential Therapeutic Agents for Triple-Negative Breast Cancer. This is a full snapshot of my poster, and within the next few minutes, I will break it down into its smaller components so that you can get a better glimpse of it. Everyone in some way knows someone who is impacted by cancer. It's such a common disease nowadays, and breast cancer is one of the most common types of cancer in women worldwide. One of the deadliest types of breast cancer is triple-negative breast cancer, or TNBC. The triple negative means that it is lacking in 3 receptors that most anti-cancer medicines target in order to stop the spread of cancer. And because of this, TNBC has very few treatment options. When plants, such as peanut, are stressed, they produce these defensive compounds called prenylated stilbenoids. And we have found that these stilbenoids have properties such as anti-cancer, antioxidant, anti-inflammatory, and more. The focus of this research, however, is to look at the anti-cancer property of these prenylated stilbenoids. My hypothesis is that the prenylated stilbenoids arachidin-1 and arachidin-3 will induce more apoptosis in TNBC cells than the non-prenylated stilbenoid resveratrol. Apoptosis is essentially the death of cells. In this research, my objective is to compare the apoptotic effects of arachidin-1, arachidin-3, and resveratrol on TNBC cells using flow cytometry. Flow cytometry is a technique that helps to detect some characteristics of cells. Future studies conducted would use different assays or tests to gain more results. In order to produce the stilbenoids for testing, I treated peanut hairy roots with 4 different elicitors or chemicals to stress the roots and make them produce their defensive compounds. Hairy roots are an immortalized plant system, so they will stay as hairy roots forever and will produce the same compounds as plants do. Once the stilbenoids are produced, they are separated using semi-preparative high-performance liquid chromatography as shown here, and then we can choose our stilbenoids for testing. Arachidin-1 and arachidin-3 are called the prenylated stilbenoids due to this extra tail added on that resveratrol does not have. The stilbenoids are then added to the TNBC cells in different concentrations, and then I can run tests on them. Using a flow cytometry assay, the number of cancer cells in each stage of mitosis can be seen. 3 days after treating the cells with the compounds, arachidin-1 and arachidin-3 have more cells doing apoptosis or are in the G0 phase where the cells are not growing compared to those cells treated with resveratrol. At lower concentrations of these compounds, this pattern is more easily seen. From my results, I can conclude that arachidin-1 and arachidin-3 at lower concentrations induce more apoptosis in cancer cells compared to resveratrol. The reason for this is the extraperennial tail that arachidin-1 and arachidin-3 have, but that resveratrol doesn't. This small extra piece results in a higher anti-cancer property. This poster was presented at the Create AState Symposium, and my research is supported by the USDA and the Arkansas Biosciences Institute at Arkansas State University. Thank you so much for listening to my poster presentation.